Hey guys, this is Mutations, the Mutation Notes Part 2, and we're just going to do a recap on slide 9 here, where you can see I've written in the word somatic cell, so somatic cell should go in here, and gametes should go in here. It's just a synonym for body cells and sex cells. You should know these, especially going into the next chapter. I'll be talking about gametes consistently, and I won't be saying the sex cells. I'll be talking about gametes, and you should know that those two are the same thing. Somatic cells are body cells. Any somatic cell is any cell that is not sperm, an egg, or pollen. That's a somatic cell. Okay, moving on to the next slide here. We have gene mutations, and with this gene mutation, specifically, we're talking about a point mutation. A point mutation means that if you had a strand of DNA, and we'll say at this position here, I was to cut that particular guy out. All right, so that's a deletion. All of these would then slide over, and that would be a frame shift. So at that point, there was a mutation. Another type of mutation would be at this point, I stick in another base that wasn't there to begin with, and that pushes all of these things over to the right. Or I could take this letter out and substitute it with another letter. So maybe there was an A in there to begin with, and somehow that got taken out and replaced with a G, which would then code for a totally different amino acid. So that's called a point mutation. It happens at a single point on the DNA. Okay. This next one is called a substitution. So this point mutation here is where we take out a single base and we stick in another base. And you can see here it goes GAT, CTC. You notice I'm doing it in threes, AGG, and CTA. So over here we've got the same GAT, and we've got the CTA. Like, wait a minute, CTA. This should be CTC. What happened was this C got taken out during like a replication, DNA replication, and the spell check didn't catch that this this A became a C, and it kept it in there. Once it keeps it in there, RNA is going to code for what this has. RNA is not going to come in and try to code, in this case, A with a U. It won't code a U. It waits and codes off of what the DNA has. If the DNA has a C here, it's going to code a G. It was looking for an A to code a U, but it's not there. So you can see at this particular point, it changed that amino acid from GLU here to ASP over here. Okay? And this mutation is called a frame shift mutation. This is what I was talking about earlier when you insert or delete a base. And the example here is a deletion of a base. You can see right here at this point, it should have been AGG. So right here it should have been GAT, CTC, AGG, and so forth. But that G has been obliterated. It's taken out. So it would go CTC, AGC, TAA. You can see it's frame shifted over to where that next base over becomes the actual part of that triplet. So why doesn't this base here change? Just because this one here is CAG, this one's still CAG. Now sometimes it will happen and everything is okay. Sometimes the base will change and it still will code for the same amino acid, and that's because of wobble. You guys got to remember on the third base, sometimes you wanted to code for like CAG, but it changed to CAC, and you wonder why it's still coding for serine. The reason why is on that third base, a lot of times it will either be like A and T, I'm sorry, A and U and C and G, and they all code for that one amino acid. So that third base is where we get your wobble from, never the first or second base. All right. So chromosomal mutations tend to have a big or large effect. So when you mutate a chromosome, instead of mutating a specific gene, say this is a chromosome here. I'm talking now a huge chromosome. And instead of having one gene, we'll say every single one of these little circles represents a gene. And instead of in that gene, you're manipulating one little sequence of bases in here that affect that particular gene, this one guy right here, you're affecting all of these. Say this section right here loops on itself and reverses. So that this strand, instead of, say, if you numbered it 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, in sequence now it goes 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Is that the same? Is it going to code for the same genes? The answer is no. When it tries to line up with its homologous pair, remember you got one set from mom, one set from dad, and say dad's messed up, and it tries to match up with mom's isn't going to know what to do with it because it's not in the same sequence. It has to be in the same sequence. It's like trying to take two zippers that are different, have different teeth. And when they try to mesh up, they're not going to mesh up right. Okay, so that does create huge problems. All right, so the next slide here. We're talking about chromosomal mutations can affect or may affect many genes. And we're talking anywhere from just a few to several hundred. So many genes. The more it affects, the less likely that little guy will be viable. In other words, would be able to survive and carry out life. It does talk about chromosomal mutations may occur during crossing over. And again, crossing over is where you have one gene from mom, one chromosome from mom, one chromosome from dad, and they're the same chromosome. So for humans, we have 23 of these pairs. And they actually number them off 1, 2, 3, 4, 5, 6. So chromosome 1 that you got from mom has to line up with chromosome 1 that you got from dad. And these will then cross over. What I mean by crossing over is parts of the chromatids, so these parts here, these little legs, will cross over. But they are supposed to cross over evenly so that half will go to one chromosome and the other half will go to the other. When they do not do it evenly, such as this, you have gene duplication. And gene duplication can result in massive errors. That's a huge chromosomal error. So because now when, say, this one goes in meiosis and becomes a sperm that fertilizes an egg, and so the egg will have the mother's genes in it. And when this gene tries to line up with the mom's gene, it's not going to be able to do it. And there's going to be a huge problem. Okay. Whoa. There we go. All right. So this one here is translocation. Now, translocations are by far the worst of the chromosome mutations. Translocation is where you would have like chromosome 1 and chromosome 17, and they're supposed to match up here at the centromere. But what happens when this centromere hooks up with this centromere, and then they undergo crossing over? The problem is you're going to have material that is in the wrong place. So when, again, moms, or the females, I shouldn't say moms, the female's chromosome matches up with the male's chromosome, and it should be number 1 should match up with number 1. Number 17 should match up with number 17. It's not going to match up right because number 17 will have parts of chromosome 1, and chromosome 1 will have parts of chromosome 17 on there, and it won't match up. It's like this one over here is trying to figure out what color hair this is, and it's looking over here and it's going, this is the size of their arms. It's like, what? The two are totally different, and when it doesn't match up, it doesn't code. If it doesn't code, it doesn't form the actual proteins that are going to be expressed later on. So that is major problems. Okay, so chromosomal mutations. So up here, this should be chromosomal mutations. Involves changes in the number and structure of chromosomes. And these are the different kinds of chromosomal mutations. You have deletions. You have chromosomes that can duplicate themselves. You have an inversion. This is where it goes in like a numbered order. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then that flips. As it flips upon itself, now it goes 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. And it tries to match up to the mate. So the females one, say this is the males that got inverted. It would try to match up with the females' same number of chromosome. So we'll say this is chromosome 13. And it will try to match up with the chromosome 13 of the female. It won't be able to do it because they're out of sequence now. Translocation, again, this is where you have one chromosome that is a mismatch with a different chromosome. And the last slide here, I want you guys to answer the questions here. Whoops. So here you have some gene mutations do not affect the phenotype. That is the outward appearance. So phenotype is outward expression. Mutation may be silent. In other words, it doesn't have to affect anything. It's mutated, but it doesn't show that it's actually mutating anything. Some substitutions can still result in the same amino acid being put in a protein. Why? If you need to figure this out, go back to the wheel or the codon chart that I showed you in the Jamboard problems. Go back to those, look at all of those, and you'll notice that some of the DNA sequence, even though it's one thing, it can code for this. And this other thing can code for this. So go and look at that wheel. There's several of them on there that will code for the same amino acid. And if it changes, mutates from this to this, and it still codes for the same amino acid, that can explain why. All right. Here you have a mutation may occur in non-coding region. Is that region an intron or an exon? Circle one. Okay. And the last one here, a mutation may not affect protein folding or the active site. Okay? And that's just a statement. You don't need to do anything with that one. So I hope this helps. I'll be going over all this later on in class, and we'll see you later. If you have any questions, talk to me in my office hours.